All right, today we're going to talk a little bit about meiosis as it pertains to inheritance and applied genetics for our biomedical science class. So you're going to open up the inheritance, mutations, and genetic disorders notes, which start like this, and then we'll do this first portion here on meiosis. So here's our essential questions for the unit. And where we're focused for this video is just here in this second question, right, which is how does an organism generate cells with the appropriate amount of genetic material for sexual reproduction? And again, this is sort of part 2 of our applied genetics, where we're still just sort of building up background info to get to the really practical stuff that's going to come here as we continue moving and making our way through. All right, so hopefully you have had a chance to check out the video about molecular genetics, which talks to you about what DNA is, what RNA is, how a cell copies its DNA, and how a cell converts DNA into RNA and then into protein. And so that definitely is sort of laying a foundation for all the other stuff that we're going to do. And we've already started talking a little bit about inheritance, right, or heredity, which is the passing on of traits from parent to offspring. But first, before we even go any further into that whole inheritance piece, we want to ask this question of how much of us is actually genetic and how much of it is actually experience? Now, a lot of times, particularly in psychology, they call this the nature versus nurture debate. And the question becomes, when we look at a trait in a person or an organism, how much of that trait is because of the genes their cells are carrying? And how much of that trait has more to do with maybe environmental factors or things like that? So first, think about your build, height and weight. We're pretty confident that there is a hereditary component to this. So tall people tend to have taller kids, and thin people tend to have thinner kids, right? But there's definitely no question, no doubt, that this has to do with your surroundings as well, your behavior, right? Your diet, your nutrition, how much you exercise can play a role in how tall you grow to be when you're a kid, and certainly throughout your life, how much weight you carry. So almost every trait actually has both of these pieces, right? The nature piece, the genetic piece, and the nurture piece, that experience piece. Kind of a fun example here are Siamese cats. Now, Siamese cats are very specific. They have this certain coloration with the dark nose and ears and paws and tail. This is a very good visible example of nature and nurture. So they have genes that code for an enzyme that makes their fur dark, but that enzyme only works at temperatures below core body temperature. So there's a genetic side, right? The fact that they make this enzyme that makes their fur dark, but it only makes the fur dark where it's cooler. So the only place you're going to see that is on the extremities of this cat and not at the core part of the body. And what's cool is you can shave hair from a Siamese cat, artificially cool that shaved area. So maybe you use an ice pack or something like that, and hair will actually grow back in darker. So it's a neat example there. Now, a lot of scientists have tried to figure out how we can tell the difference in humans between something that's more strongly genetic or hereditary and something that's more strongly environmental. And one answer they've come up with is to study identical twins. So here's some pictures of a couple sets of identical twins that were in my class a couple of years ago that you could potentially recognize these guys. What we found is if there's a trait that is more often shared in identical twins, it's probably more genetic. However, if there's a trait that's not shared by identical twins, it's probably something that's a little more environmental. And scientists have found some interesting stuff. They've been able to tie some certain types of cancer more to your genes because of studying identical twins. They've shown that obesity might be a little bit more genetic than we used to think. And they've even shown that a tendency to suffer from substance abuse dependencies. So things like alcoholism, things like drug addiction might also have a fairly strong genetic component to it. Now, inheritance, we already really kind of have started talking about Punnett squares in our classes this year, but inheritance goes back to Gregor Mendel, known kind of as the father of modern genetics. And it is from Mendel's work that we kind of led down a path to Punnett squares. And so, I kind of will take for granted that you vaguely remember the history of Gregor Mendel from freshman year, and we'll focus much more on the practical in biomedical science. The question that we need to talk about today is, where does this genetic material come from? Now, as humans, we reproduce sexually. And recall that all this means in nature is that you have, two cells that come together to form your new offspring. That's what sexual reproduction is. So when we talk about sexual reproduction, we're just saying two cells come together to form your new organism called a zygote. Now, these two cells are called gametes. And so what you do is you get one gamete from each parent, right? And you're familiar with these things already, right? The female gamete is called an egg. The male gamete is called a sperm. And when these two gametes come together in fertilization, you form a zygote. So here's an example of what eggs look like. Here's what sperm look like. And these pictures are sort of to scale with each other. So here's an image of sperm attempting to fertilize an egg in a mammal. Now, for us specifically, we have 46 chromosomes arranged in pairs. And you might have recalled that from previous biology class too. So humans have 46 chromosomes. We say that we have pairs because you get one set from your mom and one set from your dad. So in other words, half of each pair. So you're going to get this set of maternal chromosomes and this set of paternal chromosomes, which then come together as a little pair. And you will have 23 pairs or 46 total chromosomes. Now, to give you a little terminology review here, an organism, or just a single cell that has a paired set of DNA, is called a diploid organism. And diploid is represented by the function 2n, basically just showing us it's pairs, right? Organisms can have any number of chromosomes, but if it is a diploid organism, it's always in pairs, so it's always an even number. And so that's what 2n represents, where n is any number, and so if we say 2n, it'll always come out even. Now, a cell that only has a single set of chromosomes is called a haploid cell, right? And haploid, we say, is n. So whatever your haploid number would be for an organism, if we double that, that gives us your diploid number. Now, when we talk about diploid organisms, like you and me, we say that you have paired homologous chromosomes. Homologous meaning almost the same. And that's because you get a copy of a chromosome from your mom and you get a really similar copy of a chromosome from your dad. And you do that for each of your 23 pairs. Now, those will have a slight difference from each other. That's why they're homologous and not identical, right? And those slight differences are what account for the variety that we see in you, right? So you have these pairs of chromosomes, one from mom, one from dad, that are really similar to each other. Now, early scientists, at least in this field, had sort of a problem to figure out, because when they looked at human cells, they found that they had 46 chromosomes. And if you just took any human cell, right, and combined it with another human cell in fertilization, you would end up with 92 chromosomes. And that just doesn't work, right? We need our new offspring to be the same as their parents in terms of their number of chromosomes. In other words, the parents are both 2N. We need the kids to also be 2N. So there's a very logical explanation, right? The logical explanation is that the cells we use in reproduction must only have half the number of chromosomes. So in other words, we're going to have haploid cells to be used in sexual reproduction. So what we talk about in, again, an organism like a human is that the vast majority of your cells are diploid with 46 chromosomes. And we call those somatic cells. But at one place in your body, you can form a gamete or sex cell, which is haploid and has only half the number of chromosomes. Now this slide is a little bit silly with these little married tabs on here. But this is a picture of frogs mating. It was in the old textbook that we used for this course. I'm not real sure even why it was there, which is why I thought it was kind of silly. But these frogs would be like humans, right? They release eggs and sperm. And in fact, this black you see is the eggs. So the female actually releases it from her body too. And those come together to form your new organism, or a tadpole in the case of frogs. Now this is the typical life cycle of humans and most animals, at least all animals that sexually reproduce, right? We are a diploid organism that, through meiosis, that we're about to mention deeper here, can create gametes, and those gametes are haploid. Then the gametes come together, you get your new organism or your offspring, and it will develop into a diploid adult, and we start the cycle over again. Now the question becomes, how do we make these cells with half the number of chromosomes? And the answer is, in some ways, we do cell division twice, right? So we learned about mitosis a while back, and meiosis, as you might recall from freshman biology, is a really similar process. But instead of just doing one round of cell division, we're actually going to do 2 rounds of cell division. Now, I really like this picture. This actually comes from the freshman textbook. But the one thing I want to point out is that we always start with interphase, whether we're talking about mitosis or meiosis, which means that, we're always going to go through G1, S, and G2, which means we're always going to duplicate our DNA. So no matter what type of cell division we're doing, mitosis or meiosis, we're going to generate duplicated DNA and then go from there. Now, meiosis involves both meiosis I and meiosis II, 2 division stages. And for the most part, they are just like mitosis. You've got the same stages within. You've got prophase, metaphase, anaphase, and telophase. But there's just a little bit of difference in how we do this. All right, so for prophase 1, we are going to see our homologous chromosomes moving around as pairs. And we call these homologous pairs, right, of duplicated chromosomes. So instead of just a single X-like chromosome moving around in the cell, we're actually going to see these pairs of chromosomes. And the one really important thing that we'll come back to more in a minute that happens during prophase 1 is we get what's called crossing over. So these X's come together in what's called synapsis, and then they will actually exchange a piece of their DNA. And again, I'll have a slide about that here in a minute. Now, then we go to metaphase 1. Metaphase 1, still you can remember midline for this. But now they're going to line up on the midline as tetrads, which means instead of single file, they're going to line up in, 2x2 lines along the middle of the cell. Now then we go to anaphase 1. Again, you can remember apart, but here instead of splitting our X in half like we did in mitosis, we're actually just going to separate our X's from one another, right? Opposite directions. And then we have telophase 1 and cytokinesis, where we would split this thing into two separate cells, right? Each carrying a single set of duplicated chromosomes. Now this takes us to meiosis II. And the interesting thing is that meiosis II is going to look just like mitosis. You're going to have prophase, where the nucleus goes away, the spindle forms, and the chromosomes kind of attach. You're going to have metaphase 2, where the chromosomes line up on the midline. You're going to have anaphase 2, where those X-like chromosomes will split right down the middle, and we end up separating them out. But again, this is sort of the second go-round, right? So we had meiosis I, where we split into two cells, and now each of those during meiosis II is going to split into a cell. And what we'll end up with is this right here. Right? We're going to end up with 4 cells that are haploid, and, meaning they only have half the DNA, and they are going to be genetically unique from each other. Let's go back to this crossing over idea for just a second. So crossing over, pictured I think pretty well in this image right here, is also referred to as genetic recombination. All right, and this happens during prophase 1. The homologous chromosomes come together, and 2 of these non-sister chromatids, so ones that aren't identical, so in this picture we got an orange and a red, they're going to cross over a little piece of their chromosome and actually exchange it, like you see here in the bottom of the picture. This leaves us with new, unique chromosomes that have not existed before. Right? These are a unique mixture of your maternal and paternal DNA. And so then these chromosomes get passed on to your children. So what's the point we're making here? This is why you are not identical, probably, unless you are twins, to your parents or your siblings, because you are getting some new, unique, kind of shuffled up mix of the genetic material thanks to genetic recombination or crossing over. Now, we call this genetic variety or genetic variation that occurs in sexual reproduction, and it's really important, right? Variety in organisms or variety in populations allows for evolution by natural selection. And we'll talk about evolution more later this year, but it is really important that we have this variety. Now, this slide is a really good slide to study, and this is our last slide about meiosis. You basically want to know, more so than a really in-depth, step-by-step understanding of meiosis, you want to know these things. What's the point of doing meiosis, and how does meiosis differ from mitosis? So, the purpose of meiosis is to reduce the chromosomes to the haploid number. So, in other words, cut the chromosome number by half. Provide genetic variation, which happens during crossing over. And ensure that the cells you get have the correct number of chromosomes to be used in reproduction, right, to form a new organism. Now then, you definitely want to be familiar with this little table or chart over here, where we compare the results of mitosis and meiosis. So remember, mitosis gives you 2 daughter cells that are diploid, and they are identical to each other and to the parent cell, whereas meiosis gives you 4 daughter cells, which are haploid, so only half the DNA, and they are genetically unique from each other and from the parent cell. So that wraps up our meiosis review. In class, we'll continue digging deeper into inheritance and how we use this from a practical perspective. Thanks for listening.